So next we have the native perennial crops, the next generation panel. We have three panelists. We got Tom Molnar, Jeanne Romero-Severson, and Emily Warschefsky. So here we'll cover topics on domestication, breeding for disease resistance, use of wild crop relatives, and what to expect from the hazelnut breeding pipeline in the eastern US. I'll quickly reintroduce myself before introducing the panel per Hannah's request. I'm the tree nut breeder in the Center for Agroforestry. I introduced my work on Chinese chestnut earlier today, but I also collaborate with the Hybrid Hazelnut Consortium, which Dr. Molnar is a part of, and carry forward Eastern black walnut breeding schemes here at the center for high kernel percentage. Now we'll introduce the panel, Dr. Emily Warschefsky, who just joined us here. Thanks for, I think you're at PAG, so thanks for fitting this in while you're there. Emily is an associate scientist in the William Brown Center for Ethnobotany at the Missouri Botanical Garden. Her research focuses on the domestication and evolution of crops and emphasizing woody perennial species. Emily's recent research includes population genetic and phylogenetic studies on mango and its wild relatives, the coordination of international field trials of interspecific hybrid sunflower lines, and examining how genomic structural variants are impacted by domestication. Emily is an organizer of the North American Fruit and Nut Tree Working Groups, which focuses on building collaborative conservation networks for the in situ and ex situ conservation of crop genetic resources. Next is Dr. Jeanne Romero-Severson, who is a full professor at the University of Notre Dame. She's an expert on the qualitative genetics of stress resistance. Her genetic and genomic discoveries in natural and structured populations include landscape genetic and chromosome architecture of native oaks, the process and impacts of naturally occurring interspecific hybridization, and more. In 2020, Jeanne was named as a fellow of the American Association for the Advancement of Science, and her ongoing projects include a collaboration with the United States Forest Service, collaborating to develop emerald ash borer resistant green ash using traditional breeding methods, as well as metabolomic and genomic strategies. And then earlier today, we introduced her contributions to the multi-state Chinese chestnut initiative to develop ancestry informative genetic markers. Last but not least, Dr. Tom Molnar is an associate professor in the plant biology department at Rutgers University in New Brunswick, New Jersey. He obtained his PhD from Rutgers in 2006, and today is responsible for the Rutgers program dedicated for research and breeding of hazelnuts in woody ornamental landscape plants. The main focus of the hazelnut program is to develop widely adapted trees resistant to the fungal disease Eastern filbert blight, the primary limiting factor of hazelnut production in the eastern US. Tom has been working on this project since 1996 and has traveled throughout Eastern and Northern Europe, Russia, Central Asia, in search of new germplasm for use in breeding. As a result of these efforts, and with close collaboration at Oregon State University, many new resistant plants have been identified, and the first new cultivars were released from Rutgers in 2020, representing a big milestone for the program. So I want to say thank you to each of the panelists for joining us today, and we'll start with Dr. Warschefsky.
Thanks so much. I'm really excited to be part of this session. I think the Center for Agroforestry is doing some really amazing work, and I've been excited to hear other folks talking throughout the day, and look forward to the rest of this session. But I'm going to start us off, I think, with maybe a slightly broader perspective before the other speakers, I think, will zoom in a little bit on some specific topics. So I'll be talking about the domestication of native perennials, and essentially trying to sell you all on the potential of some of these North American tree crops. But really, I'll be talking kind of separately about domestication in trees, and then some of the opportunities we have with some of our native tree species in North America. So thanks, Ron, for that introduction. You all have a little knowledge about what I work on and have worked on so far. Let's see. So the first little topic I wanted to cover was just a basic discussion about tree domestication and how it works and why. So it's a little different than the domestication process in a lot of our crop species, which tend to be annual species like grasses. And there's a number of reasons why it's different. But in general, a lot of the conversations in the domestication process involve this discussion about genetic bottlenecks. And so when you take a wild species and you start domesticating it and turn it into a traditional crop, there's a heavy selection process that happens. And during that process, you tend to lose genetic diversity. And then, in the last 100 or so years, as many of these traditional crops have been improved and turned into modern crops, and we've had more industrialized agriculture, in many of our major crops around the world, we have lost a significant amount of genetic diversity. And this is a real concern for those species. And so what we're learning, though, is that in tree crops, they act a little differently during the domestication process. And this can just depend on certain factors of how they're cultivated and some of the biology of these species. And so some of my work, as Ron mentioned, focused on mango and the domestication of this crop, which originally was being cultivated on the Indian subcontinent. And then over the past few hundreds of years has made its way all across the world. And my research into mango helped to show that for some perennial species, this domestication process doesn't have the same impact on genetic diversity. And so in the case of mango, we didn't see these genetic bottlenecks as it was distributed, as it came from India into Africa and then into the New World. We didn't see a loss of genetic diversity. And so in some ways, that's a really good thing. And this holds up across many woody perennial crops in trees. In fact, most of them, rather than losing a significant amount of genetic diversity, are able to maintain almost the same level of diversity as you see in sort of the wild populations. But it's important to understand kind of how that's happening and why. And a lot of that comes down to the biology of tree species, right? So, some of these things are really obvious. Trees live a long time. And so that means that the selection process is happening at a slower rate, right? Trees are very tall. This has implications in terms of thinking about the population size of these tree species. Pollen dispersal in many trees can happen over kilometer distances rather than just a fairly short area. There's also the fact that many of our tree crops and tree species have to outcross. So you're not creating these inbred lines that you see in annual species. And this means that you're maintaining higher levels of genetic diversity within single individuals, so higher levels of heterozygosity. There's also some evidence that shows that even at the scale of mutations, trees evolve more slowly. So not just due to their long generation time, but also, at the population level, because there's a lot of gene flow happening, and then also at that individual level of mutations. And so for all of these reasons, it kind of indicates to us that the tree domestication is happening kind of at a different scale, both in terms of time and in terms of geography and physical scale. So tree domestication seems to be a much longer, slower process. It happens perhaps even at the landscape scale rather than in an individual field or a small region. Oftentimes, because trees have to outcross, you get a lot of cases of hybridization between species, which again can be a really useful thing. And it sounds like we're going to hear some about hybridization between species in this session. And it also has this unique feature where, because of these high levels of diversity within an individual, because we can't create true breeding lines, a lot of trees are grown clonally or vegetatively to kind of capture these genotypes. And some of our knowledge about how tree domestication works is actually coming from really interesting research looking at the domestication of Amazonian forests. So looking at archaeological evidence for how people historically domesticated these forests at this landscape level, which is just really fascinating research to me. All right, so kind of giving you that really brief overview of where I come from and how I think about tree domestication, I want to talk to you about kind of the potential of trees in agriculture, in agroforestry. I assume most of you are already on board with this idea since you're here at this symposium. But there's an increasing, thankfully, an increasing acknowledgement that trees can and should play a really important role in the future of sustainable food systems. And so I just want to acknowledge that this is becoming a more popular in the literature at the scientific level and also in policy. And it makes a lot of sense. I always like to show this slide in my talks because I think a lot of people forget that tree crops are actually pretty economically important at the global level. Of the around 145 major crops that the Food and Agriculture Organization keeps track of, about a third of those come from woody species. And roughly a fifth of the world's most important crops, and that's in terms of both production value and production kind of quantity, come from tree crops. So you can see some of the examples of these really economically important species here on this slide. There's also some things to consider when we're thinking closer to home and thinking about new or quote unquote new tree crops. A lot of times what we might be talking about in sort of the more agricultural side of things as new, really, we obviously have to acknowledge these have probably been used as traditional food sources in many of the regions that we're looking at, especially here in North America. We have a lot of species that indigenous cultures have made use of for many, many years. But some things to consider when we're talking about tree crops is that many of our sort of newest crops or domesticates at the global level are woody species. So think things like kiwi and macadamia, and even more locally blueberry and cranberry. Relatively young crop species in terms of sort of turning them into industrialized things, and they're woody species. Trees can also provide a lot of really interesting resources. So of course they provide things like fruits and nuts, which is what a lot of us focus on for good reason. But there are trees out there that you can eat the foliage of them. Trees provide us with spices, some of our most important beverages like tea and coffee. And so there's a lot of potential for a diverse suite of crops that can come from trees. And thinking about the trees specifically in North America, one of the reasons I'm particularly interested in these species is that not only can they be useful in and of themselves, but they can also be important genetic resources for existing global crops. They can be used directly in breeding programs because of this ability to hybridize with these crop species, and they may have important tolerance to environment or disease. But oftentimes they can also be used as rootstocks, which I think is an undervalued potential for these species. So most of our tree crops are grafted, and rootstocks have the ability to confer a lot of really important characteristics. And this is something I kind of gained interest in as a graduate student as a side project, but I'm excited to see that more and more there's talk about this. So I'm at a conference right now, and I heard a wonderful talk about how Mars, the chocolate company, is actually doing selection on cacao rootstocks and breeding rootstocks for cacao. So this has a lot of potential as well. And so I think this makes the case as to why, our wild relatives that we can find in North America are particularly valuable. And so that's the last bit of what I want to talk to you about is, as Ron mentioned, I'm part of a working group that's focused on the collaborative conservation of North American fruit and nut trees. And I just want to give you all a little bit of an overview about what this is and why we're doing it, and kind of where we're at and where we're going. So this is work that I'm participating in, and it's being organized and supported by Botanic Gardens Conservation International and the US Botanic Garden in collaboration with San Diego Botanic Garden. And so this work started out from a paper that came out in 2020, looking at the conservation status of crop wild relatives in North America. So this was all wild relatives of important crops that can be found throughout North America. And so this heat map is just kind of showing the distribution of those species. What I found really interesting is for those of us in Missouri, you can see Missouri is part of this area of high density of crop wild relatives. And it's in a region where you might not expect there to be critical biodiversity. But what popped out to me when I was looking through this paper is this figure that's on the right. The column in red is the taxa that were classified as urgent priority for conservation. And I parsed it out and saw that in this column, woody species are heavily overrepresented. So, there's far more woody species that are in need of urgent priority conservation action than you would think just based on the percent of these woody species. And so this set us off on a task to form some collaborative conservation goals for these woody tree species here in North America. And these are kind of the three goals that we're starting out with for this working group, which is to establish and foster a network of experts, to identify and prioritize the species of greatest concern, and then to develop some ex situ conservation collections of these species. And ultimately, we're hoping that these ex situ collections will inform, they'll serve as conservation repositories, but also be useful for crop research. So we have now we've been together for about 2 years, the North American Fruit and Nut Tree Working Group. And in this working group, our target folks that we're trying to work with are folks from botanic gardens and germplasm repositories, land managers, researchers from universities. We're also very interested in just including anyone who is interested in thinking about conserving and using these wild tree species in North America. And we have focused so far on 10 genera, 5 nut genera and 5 fruit genera. That includes about 90 species total. what we started off doing was just a survey to figure out, what do people have in their collections already? So in botanic gardens and gene banks, how many individuals of each of these species do we have in ex-situ conservation collections? And so this is just a figure to show you, in some cases we have a lot, in some cases we don't have any for some of these species. And then we've used that data to examine the status of conservation for each specific individual taxon within this study, right? So we have, for example, Juglans californica, a Juglans that is native only to, Southern California. And in this map, you can see the green is this predicted wild distribution. And then the purple and pink dots show the origin of where we have collected material. And so that means that either a gene bank or a botanic garden, depending on the color, has an individual in their collection from that particular location. So the reason we want to do this is it gives us an idea about, are there parts of the regions for these species that we may need to collect additional individuals from. And in this case, you could see sort of there's this northern extent of the range, the southern extent of the range. There's quite a few gaps in our existing collections. So this gives us a starting point to know kind of where we should prioritize future work. And we've done this for every species of these 90 taxa. If anyone is interested in the species level data for this, or working with us in this working group, please feel free to reach out to me. I will get you involved. I will get you the data sets. We're happy to have as many collaborators as we can. And so, like, as I said, we've done this for all of the species in this group. So you can see here, this is another Juglans species. The entire southern extent of the range has not been conserved. And so it's potentially at risk of being extirpated, especially somewhere like California, where there's lots of development going on. And so I'm going to stop now so that we have time to hear from other speakers. But I just wanted to end with this slide and mention that, I think what I've presented here, I have in a framework of, including what we do at the level of crop diversity. I understand that's within a context of human diversity and ecological diversity that are all really critical to what we should be thinking about in terms of the future of domestication. And so I'm very thankful to see that, this symposium has included a lot of talks that address all three of these different topics. So thank you.
Thanks, Emily. Next, we'll go to Jeanne.
All right. Thank you for inviting me. This has been a wonderful symposium. I very much regret that we don't do these things in person, but I understand why. I have been given the fun and rather onerous task of explaining breeding for disease resistance in trees in 15 minutes. So I'm going to tell you everything I have learned about breeding for disease resistance in anything and in trees in 15 minutes. Okay, so when you're breeding for disease resistance, let's assume first that you're on board with the concept of breeding for disease resistance. I represent the approach here as a series of triangles. First of all, a disease or an insect or even a abiotic stress consists of this, what we call the disease triangle. There has to be some sort of stressor. In this case, let's call it a pathogen. There has to be a host that is that can be stressed or killed. And there has to be an environment that influences the host and influences the pathogen in such a way that disease or destruction can develop. Okay. In order to effectively breed for disease resistance, this pathosystem must be understood. Okay. At least understood enough so that you can start to move to triangle number 2 to characterize the phenotypes of your susceptible hosts. Okay, and when I say host here, I don't mean one tree in your orchard. I mean a collection of trees that you are interested in. Okay. When you characterize the host response to this stressor, this pathogen, you can design a breeding program given what you see in that host phenotype, and then these two things mutually reinforce each other. What I'll discuss minimally and only in its appropriate context is how this activity can lead to the characterization of specific genetic and metabolic mechanisms that in theory, can help you to characterize the host phenotype. And why do you do all this? You do this so that you can be really good at plant breeding. Here's what you need to do to be really good at plant breeding. 1, pick the best parents. This is hard. That's what this is about. 2, keep track of your stuff. Okay, I cannot emphasize this enough. And 3, have quantified long-term goals. Let me give you an example. Let's say I want to breed American chestnut for American chestnut. Let's say I want to breed Eastern hazelnuts to be European hazelnuts to be resistant to Eastern filbert blight. Okay, well, I must have a clear idea of what that resistance looks like. I need to find some source for that resistance. Might be in the European hazelnuts, but it's most likely to be in the American hazelnuts. I hope I'm not stealing too much of your thunder here, Tom. You can do this. You can do this for your whole life and for years, and it will be completely fruitless if you don't keep track of your stuff, because what will happen? Tom Molnar has worked his whole life to develop these beautiful hazelnuts. He dies when he's an old man and has left behind no records. What's going to happen in the fullness of time is 1 or 2 of those cultivars may be used, and all the rest of the really promising stuff will probably be plowed up and houses planted on it. So much wonderful plant breeding effort, especially in perennials, is lost because of this failure to keep track of your stuff and have good records. And now that we have digital systems, no more excuse for that. Okay. Right, resistance breeding in woody plants. This sounds easy, but it's not. So characterizing the host phenotype pathosystem can take years. For example, in my pathosystem, resistance to emerald ash borer, we need to know what that phenotype looks like in an orchard, in a forest, in a greenhouse, in a growth chamber. We need to be able to recognize the difference between a susceptible that we don't particularly want and something that's more resistant. In a long-lived perennial organism, there can be an influence of the age. There can be an influence of whether or not it's grafted, depending on the pathosystem. Some pathosystems involve the roots, such as Phytophthora, where this matters very much. There's an influence of genotype within a given species. There's a wide range of genotypes in most all outcrossing forest trees. And of course, there's genetic variation in the pathogen. I'm using one word here, biotype, depending on the pathogen or the insect. The community has different words for this. Doing this can be done in parallel with phenotyping the hosts for a resistant phenotype. Before I get any further here, let me explain about resistance and susceptible. There are very few things in nature that are just resistant and just susceptible if your species is a host at all. What you usually see, not always, but what you usually see is a continuum, or more of a continuum. A good example is the response of Chinese chestnut to chestnut blight. Chinese chestnuts get blight. Okay, the fungus is able to get in the tree and grow some, but all that it does is there are these little bark cankers here and there in the tree, but the tree doesn't die. It is only mildly inconvenienced, if at all. And we call that phenotype resistant. It's not immune. It's resistant. There are some Chinese chestnuts that maybe have a few more cankers, and some Chinese chestnuts that really don't look very well at all. Okay, so the plant breeder has to have a clear concept in his or her mind of what resistant means while you're characterizing the host phenotype pathosystem. So what we usually do here during this learning process is to screen cultivar or germplasm collections of the native species or a European or Chinese species that we know is resistant, looking for a number of resistant phenotypes. Okay, if you're working in natural populations, as we are with emerald ash borer, what you need to do is monitor hundreds and hundreds and hundreds of forest plots for years and years and years. So this takes great patience and a lot of hopping up and down with people who fund this to try to get them to understand that this is a work of years. No matter what, it's a work of years. So you got to hold on. Develop an assay for host response. It is very difficult to get gain from selection, breeding for resistance, if you don't have some sort of assay that you can use. I'm planting a whole lot of plants. I want to be sure that each of those plants actually gets the disease. And plant breeders are very creative people about developing these assays. I don't know, Tom, if you're going to describe to people what you do. It's really, and that's not the only system. We'll go to great lengths to develop this assay because we must have it. In nature, a tree may look very good, but it could be an escape, and you don't want that. Okay, once you characterize your pathosystem and phenotype hosts and understand what resistance means in your system, you can initiate and sustain a breeding program, which is going to take a whole nother number of years, even in a perennial crop that has a relatively short cycle. All breeding involves controlled crosses. Okay, that's the design of the breeding program. There's more breeding approaches than the back cross. There's whole books on this. What kind of breeding program you use depends on the host phenotype and what you've learned about that. But at some point, you make controlled crosses, or you can plant all your good trees in an orchard and let Mother Nature make the crosses, but some sort of deliberate strategy to make these crosses has to happen. The point of doing this is to do progeny tests, because the progeny tests tell you what the value of the parents are. Who are the good parents? Good parents are the foundation of your breeding program. And then what do you do next? You cross best by best, but understand, which is something that Greg Miller emphasized earlier in the day, that best is not one golden plant. Best is a collection of things, of which the disease resistance is one, but not the only one. And no breeding program wants to form a foundation based on one plant. It can start that way, but you don't want to continue that way. And then in the fullness of time, there's all this other stuff, more about which later. Okay. Oh, going backwards. Let's not go backwards. Okay. Here is a huge simplification of the breeding for disease resistance workflow. Okay. This applies to all crops, not just perennials. You get some sort of disease, some sort of thing that does in your trees or does something very undesirable to them. You do the process of phenotyping the hosts. You make crosses so that you get some kind of structured population. You screen for resistance with the assay you have developed to ensure that all the plants you look at have a uniform challenge. Then you replicate that. So this goes round and around for a while. You propagate the best, in your definition, resistant progeny. These progeny are now hosts, and this goes around for a while. Okay. Your ultimate goal, if you're working in a domesticated species, or if you're working to domesticate a species, is to develop cultivars, or at least develop some sort of land races with the appropriate local adaptation, with the disease resistance, while at the same time preserving genetic diversity. This is absolutely essential in perennial species. Your disease isn't the only thing that this tree has to withstand. So this is absolutely critical preservation of genetic diversity. In natural populations, the goal is self-sustaining, durably resistant populations. You do this, and in many circumstances, this works fantastically well for disease resistance. It's worked very well under favorable circumstances in row crops, in fruit trees, and some nut trees. I won't go into favorable circumstances. I'm happy to take questions about that later. Okay, now most of us plant breeders who find ourselves in academia, and all of the plant breeders in the private sector who work in high value crops, corn, soybeans, potatoes, and so on, also add on this part to their workflow, the orange stuff, the molecular stuff. Okay, sometimes the goal is to identify predictive metabolites of proteins so that we can screen for resistance more efficiently, so that we can run these cycles more efficiently, ensuring that we're more likely to get something good at the end of these cycles or while these cycles are running. We also, just for the pursuit of truth and knowledge, would like to know more about plant host, this should be pathogen host interactions. We know spectacularly little about this, even after all this time. We'd like to identify regulatory genes and effector genes. We'd like to characterize the metabolome deployment, the nasty chemicals that trees can use to kill insects, especially stem borers. All this technology here, this orange side, unfortunately, after about 20 years of doing this, this orange part of the integrated approach has had minimal impact on the delivery of resistant trees, except for DNA markers. DNA markers are the best thing ever to keep track of your stuff. Okay, the best thing ever. Whether or not they help gain from selection depends on your circumstances. But if I have a DNA footprint, a DNA panel of the tree I have, I will always be able to identify it. I'll be able to identify the parents, and I'll be able to sort out all the errors that inevitably happen, no matter how good you are and how good the people in your program are. So, other than that spectacular exception, why are we doing all this when it has had ... so very little impact? Part of it is the fourth triangle. So successful resistance breeding in woody perennials requires a long-term commitment, much longer than the 3 to 4 years of an NSF grant, much longer than the patience of, say, your dean in the College of Agriculture, a long time, years and years, extending over careers. In order to sustain a long-term breeding program, you have to have stable funding and you have to have sufficient funding. This seems so obvious, but this leads up to why we do so much omics programs. In order to sustain a program like this, you as the academic plant breeder or the private sector plant breeder or the .org plant breeder absolutely must have early success. Funders want to see that you're doing something that will move the counter towards a resistant plant or move the counter towards domestication and diversification of, say, American persimmon, one of my favorite native plants. One of the ways to do this is to be lucky and land on a single gene resistance for the plague of the day. Okay. If you can find single gene resistance in a related species or single gene resistance even in the native species, and you have a group of susceptible but otherwise very good cultivars, it takes a little time, but it's straightforward to incorporate this single gene resistance into those cultivars. You look good, your dean is happy, the growers are happy, you have early success, and this directly helps to ensure the stable funding that you need for the long-term success, to build on that early success. Omics programs also do this. If you can say, look, we did metabolomics, we're working towards understanding what the mechanisms of resistance are. This moves a counter that is a very long-term deal, but funders are impressed with this. At least in academia, they're impressed with this. Okay, this allows you to gain more funding and ensure the stability of your program. So I tell people, pursuit of truth and knowledge is a very good thing. It's one of the reasons I went into science. But in a long-term breeding program, unless you ensure stable funding, it's fruitless. So this is essential. Okay. What is long-term success when you're breeding for resistance of any kind? Okay. Deployment of durable resistance, which single gene resistance is not. Mother Nature has it out for single gene resistance. Mother always overcomes this. I don't know of any single gene resistance in perennials that hasn't been overcome in nature. This is true in row crops too. There are hundreds of stories, sad stories about relying on single gene resistance. Durable resistance is usually based on stacking up of very good single genes or dealing with quantitative resistance, which involves many genes, which is something people like me work on. Quantitative genetics is a detailed thing. All the quantitative genetics people in the world, all the senior people could fit in one small classroom. But we do it because it's worth doing, because it puts this on the landscape. This is what evolution does. Durable resistance. Okay, not immunity. The pathogen, the insect still persists, but the tree doesn't die. It's still healthy. It still reproduces. Long-term success in any environment is really helped by very successful cultivars that the growers love. You can get if the university or your .org or your US, I don't think USDA does this, but if there's a modest royalty stream from a successful cultivar, this can ensure stable funding and thus provide you the opportunity, you and your colleagues, for long-term success. This is much more difficult in the restoration of natural populations, which we're trying to do in our molasse borer, because you don't get any royalties for this and you don't want them. This is, if anything, is for the good of the cause. It's the restoration of natural populations. And so far, we've convinced the Forest Service and the USDA to just fund us just enough so we can make this happen. And then a private donor stepped up. So we're going to be able to do this. Don't rely on your private donors, though. This doesn't always happen. Okay. One last slide. There are a lot of misconceptions about breeding for disease resistance in trees. Misconception number one, traditional breeding for resistant takes longer than CRISPR, and CRISPR stands for all this stuff in this slide. Misconception number two, tree breeding is just backcrossing. Any idiot can do that. I mean, why do we need to have a breeder do that? Wrong. Tree breeding requires more money than CRISPR. Also wrong. And why is this wrong? This is the primary reason why these are wrong. CRISPRs and related technologies still require, you have to phenotype the parents, you have to make the crosses, you have to perform the progeny test, you have to maintain genetic diversity, you still have to have orchards, greenhouses, people to take care of all those plants. So what CRISPR and related technologies does is make the plant breeding process more efficient, but it doesn't necessarily make the time to success less. Okay. Last idea, CRISPR. CRISPR's only good if you know what to CRISPR. We have Arctic gold apples and non-browning mushrooms because those biochemical pathways that result in browning were worked out many, many years ago by plant biochemists toiling away at universities primarily. So it was known exactly what to do to prevent browning. In most resistance systems, we don't know what to CRISPR. Thank you, and I'm done.
Thank you so much, Jeanne. That was great. I think that it's a nice sequence of speakers we have here. So Tom, building on some of your ideas with examples from his program will be very nice.
Tom existed before I started doing this. And I very much appreciate what he's done with hazelnuts, one of my favoriteest nuts of all.
And before we jump to Tom, I'll also say I appreciated your last slide as gene editing and being a tool for, not a replacement of, breeding.
Mm-hmm.
But I will turn it over to Tom now.
Okay, well, this is actually really great to follow Emily and Jeanne. I think it really set the stage quite well. And honestly, if I had to do what Jeanne just did, it would have taken me like 5 hours. I mean, that was an incredible distillation of breeding for disease resistance. I mean, that was awesome, Jeanne. And I'm actually not really going to talk much about that necessarily or specifically. So I'm going to bring it to just some sort of hands-on, you know, what have we been doing to try to meet these goals? And I'm glad that Jeanne went through all these different steps because she basically just outlined the past 25 years of my life working on hazelnuts. And Emily really set the stage too, because we've been looking at plant genetic diversity and using plant wild relatives, the wild relative being the native hazelnut, as sort of underlying our breeding efforts. So it all connects together, and I will have a lack of details on how we go about some of this, but I think Jeanne really covered it. So it probably won't be very redundant as we go. Since we are going to talk specifically about hazelnuts, I wanted to just bring everybody on the same page here. It's really great to see that we have so many participants. I'm looking at 161. That's pretty exciting that you can have that type of a reach for a 15-minute presentation today. Right now in the United States, 99% of the U.S. hazelnut crop is grown in the Willamette Valley of Oregon. I know a number of you already know this. Just want to talk about it a little bit though. Production continues to expand. They're now at about 90,000 acres. And they've doubled the acreage in the past 13 or 14 years. It's quite exceptional how large and how fast they've expanded. But this is supported by research and breeding at Oregon State University. But as most of you know, there's also a significant desire to grow hazelnuts in the eastern United States. And when I say eastern United States, I mean just east of the Pacific Northwest. So that's going to include the upper Midwest, the Midwest, the Great Plains, the Northeast, the Mid-Atlantic. So we're just going to lump everything else, just not in Oregon. Right now, there's a lot of momentum for planting hazelnuts, just like for planting chestnuts. With hazelnuts, orchards are being planted with trees from a variety of sources. Now, these aren't necessarily clones. They could be seedlings, they could be clones. There's a lot of things getting planted, a lot of plants getting planted right now. The industry is in its infancy, but it really is expanding. And there's, like I said, there's quite a bit of momentum. Hazelnut production is on the horizon outside of the Willamette Valley of Oregon. And at Rutgers, we've been working on hazelnuts since 1996. I've had the fortunate experience to have started or helped start the program with Dr. Reed Funk when I was a freshman, and somehow just never left. I'm one of the few people that my undergraduate research project turned into my graduate research project and turned into my academic career. So it's been quite a few years, over 20 years of working on this crop now. Eastern filbert blight is the main limiting factor of production of the European hazelnut. This is caused by a native fungus, Anisogramma anomala. We can't be upset with the EFB fungus because it was here before we were. It is native to the eastern half of the United States. And what we're trying to do, or some of us have been trying to do, or historically, is grow and introduce host plant. So we're trying to grow the European hazelnut. It happens that most, not all, but most of the European hazelnuts are highly susceptible to this fungus. I'm not going to talk a lot about the details of the fungus. Just know that it's quite devastating. It kills the plants. So you really, it's not like a powdery mildew or a leaf disease that weakens the plant or makes it look ugly. It kills the plants. The other thing I want to point out also is that the European hazelnut is adapted to not only the Pacific Northwest, but really what we call the fruit belt of the northeastern North America. So you can see here within kind of this rough area I sketched out on the map. This includes parts of the Mid-Atlantic region, the Great Lakes region, really the USDA climate zones 6 and 7. They do best, the European species does best where we have moderately cold winters. They can take some cold, but also wet, hot, humid summers. If the European hazelnut is eastern filbert blight resistant, it can grow quite well and be productive in these regions. So going back to the beginnings of the Rutgers hazelnut project, finding resistance in European hazelnut, then breeding locally adapted cultivars has been the focus of the program. Today, I'm going to talk about hybrid hazelnuts, and you'll see sort of stepwise, it moves forward from just looking at the European species, because of course, we're excited about being able to significantly increase the growing range. So the early goal of the program, and I love how Jeanne kind of put this all into context, but we were really trying to identify resistance within the European hazelnut. This was the big deal here. So collaborating with Oregon State University, we screened tens of thousands of seedlings from controlled crosses. I should mention that we have collaborated very closely with Oregon State since the very beginning of our project, which allowed us to build off of several decades of breeding of hazelnuts before that time. So now we're looking at 25, 35, 45 years of perennial crop breeding that we're benefiting from as we of course step to the year 2023. So we screened control crosses from Oregon State, breeding selections, clonal plant material, and then I had the really fortunate experience as a younger person to make a lot of germplasm collections. And you can see this kind of colorful map here with a lot of red arrows. We were able to make seed collections, not looking for commercial cultivars necessarily, but locally grown cultivars all throughout the native range of the European hazelnut, and then into sort of introduced range. It's not native to Central Asia, but through the Soviet Union, they moved a lot of plant material. So we were able to go and just collect seeds of hazelnuts, bring them back to New Jersey. This is a lot of my graduate studies work, make these collections, bring them back, and then screen those plants for eastern filbert blight resistance, adaptation to our climate, and of course, then kernel quality and yield. Eastern filbert blight is native in the eastern part of the United States, and we have very high disease pressure on the farm. So to Jeanne's comment of being able to phenotype the host plant, my early years were trying to figure out my screening methods, figure out this fungus. So when I saw a plant that didn't show disease or showed only a little bit of disease, I would be confident that it's showing some sort of host response. And then we could use it in breeding. I'm going to end the story here on the European hazelnuts because I want to jump into hybrid hazelnuts where we're actually going to use our native Corylus americana. But this is a big deal for us, especially since I've been working on this crop since I was 18 years old. In 2020, we released our first cultivars from the program. And this, like I said earlier, this came through these really great collaborations we had with Oregon State University. Maybe some of you out there have seen the names of these plants: Raritan, Monmouth, Hunterdon. And what's really exciting, we didn't just release these plants. These are now being sold by nurseries and they're being planted in the ground. Over the past 2 years, there's probably at least 15,000 trees, probably closer to 18,000 trees have been sold, mostly of Raritan and an Oregon State selection, which I may have time to talk about the bees. But it's kind of neat to be in the point where you can actually go and order these trees that we've been dreaming about for a long time. Of course, being sort of an ambitious plant breeder and working with a lot of people interested in growing hazelnuts in colder regions, if we had better cold tolerance, cold hardiness, climatic adaptation, we could really expand the production range. And this is really a great time for hazelnuts or for hazelnut research and breeding. There's actually multiple projects underway in the United States and in Canada, or Ontario in particular, to explore hybrid hazelnuts, so developing them as a crop. I'm going to talk specifically about the Hybrid Hazelnut Consortium, which is Rutgers, Oregon State, the University of Nebraska, Arbor Day Foundation, and the University of Missouri. But I'm sure many of you are also familiar with the University of Wisconsin, University of Minnesota. And I should say that we're actually joining forces. We just are in initial stages of putting a proposal together with this entire group. So we're really working closely with the Upper Midwest group. But there's also multiple private nurseries that are working on hazelnut breeding and selection, and propagation and sale of plants, and really industry development. It's kind of exciting. So quickly, I think many of you may be familiar with what hybrid hazelnuts are, but these are crosses between our wild American hazelnut, Corylus americana, which I'll show a slide about that. But in general, at least the northern selections are very cold hardy or cold tolerant, and also having evolved with the Eastern filbert blight pathogen, very resistant or highly tolerant. So the goal here is really to combine the improved nut traits, the domestication traits of the European hazelnut with the adaptation and disease resistance of the American hazelnut. So there's a lot there, but the genetic resources are available to do that. They cross freely and you get viable offspring, fertile offspring, I should say. And this has actually been going on for now over 100 years, at least efforts to do so, but it's really been intermittent. It's been just a few plant breeders here and there, and just how Jeanne sort of mentioned, plants are lost, records are lost, a few plants kind of squeak through and make it to the next generation. But we do have this really interesting history of hybrid hazelnut development that started, I think the first year, the first cross ever was 1918. So we're now over 100 years of hazelnut breeding. We've had breeding progress, but no commercial cultivars released. At this point, the most widely planted hazelnuts outside of the Pacific Northwest are mostly from seed-derived orchards. They're actually highly variable in terms of commercial nut traits, but they tend to be widely adapted, disease resistance or disease-resistant populations, and really convincing of their potential. So while you can say, well, you can't really make money growing hybrid hazelnut seedlings, well, they've actually inspired a lot of people to really think about this as a crop. And then now it's, turn it over to plant breeders and others, and how can we really cultivate and make selections. But it's really, there's a lot to be said with the people that have worked on this crop in the past. The American hazelnut, it's a small multi-stem shrub. It's widely adapted. You can see this. It's kind of an old native range map. I don't know exactly where it is to date, but it goes to some very cold regions. It grows on a wide diversity of soil types, even including very sandy soils in the upper Midwest. So it's very widely adapted and phenotypically plastic, I would say. You can grow it in a lot of different conditions. And like I said, it's cross-compatible with the European hazelnut. And what we're trying to do here is really not necessarily look at hazelnut as a new crop. This is a very old crop. It's been domesticated for thousands of years, really. And there's an international hazelnut industry. There's equipment, there's markets, everything's there for hazelnut. But it's not there when you have itty bitty tiny nuts with thick shells and plants that aren't able to be harvested with existing equipment. So you have two different approaches. You can develop equipment and markets, or I'm a plant breeder, when we say, well, why don't we try to get plants that can grow all across this native range, but have kernel qualities of the European hazelnut. I don't need to invent a new machine. No offense, because I do like the ideas of the other machines. They're really great. But personally at Rutgers, we can just buy an Italian machine to harvest the nuts. We buy an Italian cracker to crack them, and then we can sell them to people as hazelnuts. So that's been sort of our goal, is to see where we can get with hybrid hazelnut development to get that kernel quality that we can sell it on the international market, but also have high quality kernels that we can sell locally, because that's really where a lot of the focus is. As I mentioned, we have this group in the Hybrid Hazelnut Consortium, and I really just want to talk about one kind of interesting component that we're doing in terms of breeding, where I think we're excited over, or I'm personally excited over. But realize we have Oregon State, Rutgers, New Jersey, University of Nebraska, representing one of our harsher climates, and then of course, University of Missouri, which is a relatively new component. But the goal here is to help secure current production in Oregon. And when I say secure, I mean help them identify durable resistance and climatic resilience. You know, as we look towards the 30, 40, 50 years from now, it's kind of worrisome what the climate's going to be like. And then, of course, expand commercial range across North America. And then the one thing I wanted to talk about is our multi-region breeding strategy with the goal of developing widely adapted cultivars. With the premise here that if we have a cultivar that can grow well in New Jersey, well in Oregon, well in Nebraska, well in the upper Midwest, or I'll say a set of cultivars, but we can identify those that are adapted in plastic where they can grow. And I see this, we have plants from Minnesota that grow really well in New Jersey. They grow well in Nebraska. Those may be the plants that can survive some of these really wacky climate fluctuations we're seeing, or these other issues in the future. So we're trying to do our best to develop plants that are going to be suitable for the long-term horizon. It's hard to know what's really going to work, but we're going to give it a shot. In terms of a breeding foundation, we'll have to give a lot of credit to Sean Mellenbacher, who worked to widen the germplasm base for hybrid hazelnuts with foresight back in the late 1980s. He worked with members of the Northern Nut Growers Association and asked them to go into their forests, their local hedgerows. I don't know where they found them, but to find American hazelnuts and send him seeds. He grew out hundreds of plants from many dozens of collectors all over the country. And what he did as a really strict plant breeder, he assessed them, he took notes on them, and he selected the best in terms of the traits he was interested in, one being bearing nuts every year, larger kernels, being able to get those kernels out of the shell or out of the husk, things like that. And he put together what I would call like a core collection. So he built a collection of less than 100 individuals. He also began crossing them with his own elite European hazelnuts. So he developed this hybridization breeding program starting in the 1990s. And when we started the Hazelnut Consortium, he openly shared. He said the Oregon hazelnut industry is not interested in these crappy interspecific hybrids. They have elite cultivars. It's like, so the university said, you know what, you can share this material, help the industry develop elsewhere. So he was so generous in sharing his plant material. He also used the early New York breeding selections and crosses, what I won't get into. But the main thing is he actually collected wild plants, he made selections, and he started to cross them with the idea, and then later using genetic markers to try to use the most diverse he can and to widen genetic diversity. He even began crossing his best F1s to make F2 populations, using genetic diversity based on DNA markers to cross wide parents. So he had this really great scheme in place. The other player in this, and I think this is really where it kind of gets interesting. And if you can picture, I have this pool of germplasm from Oregon State. Now I have this pool of germplasm to play with from the Arbor Day Foundation, where in 1996, they planted 5,000 hybrid hazelnut seedlings from the Badger Set Research Nursery in Minnesota. They grew them out, and it actually got them excited over hazelnuts. About 10 years after that, they started to work with the University of Nebraska. They put a graduate student on the project. They went through and evaluated all the plants. Don't know how she did it, but she did it. And she found the top 10 highest yielding plants over about a 5-year period. They were tracking these plants and they found the most consistently high yielding plants. So they were able to say, Tom, I've got the 10 best plants out of this planting. And here's the data to show it. And it was impressive. The yields were close to Oregon yields. So as a plant breeder, and this is just a picture, this is behind the lead lodge, it's still there. As a plant breeder, I get excited over this. Unfortunately, there was no Eastern filbert blight. So I've got their 10 best, highest yielding plants, but there's no Eastern filbert blight. They're adapted to these harsh Nebraska conditions, but are they disease resistant? I don't know. So what we did was, well, we clonally propagated those 10 best plants. We clonally propagated a whole bunch of stuff from Oregon. We brought them to Rutgers, where we probably have the worst disease pressure potentially in the world. I can be to blame for part of that, but naturally it occurs there. And we screened all this plant material under very high disease pressure. What that allowed me to do was to select the most resistant, most tolerant survivors, also select those that had the best nut quality and yields in our local conditions. So now I have Oregon, I have Nebraska. Then we start our breeding program. So here, under our New Jersey conditions, we're crossing the Oregon, the Nebraska, European, interspecific hybrid, pure American, a whole bunch of different things. We grow out these progenies. 8 years later, we evaluate them for all the traits we're interested in, and we start to make selections. Here's just a pretty picture. But what we found was, even in F1 hybrids, we are now getting kernel quality traits that are very close to the yields of, or to the traits of interest of the European cultivars. And you can see here, the one I'll point out is Yamhill, which is for the kernel market. It's got basically a 1 gram kernel, 46% kernel to shell, basically. Those are our targets. 1 gram, anything over 42% is actually a pretty thin shell. And you can see here, and of course, this took about 10 years to get to this point, but here are some of our best selections, which actually have kernel qualities that would fit in the kernel market. And they're disease resistant. Are they cold hardy? We don't know yet. Here's a picture of pure F1 hybrids. And I like this slide because this shows, we had a couple parents that floated up to the top, which tends to happen in breeding programs. Here's a pure American hazelnut from North Dakota. We're crossing it with an elite OSU European hazelnut, and we're recovering resistant plants with maybe not top of the line kernel qualities, but very close. Much, much better than most of the things that are available in that region now. Wisconsin, Minnesota, we're expecting that these will be more cold hardy than the European hazelnut. We're not really sure yet. So what do? We go through this big breeding program, trying to combine all these different plant materials from different regions, selected New Jersey. We identified the top 50 out of about 3,000 individual plants. Then we start to clonally propagate them. Now remember that these are disease resistant in New Jersey and have commercial quality kernels. So exciting from our perspective, they grow well in our climate. But what's really interesting is, well, how are they going to do when we go across the country with them? So we now have trials, Nebraska, Missouri, Minnesota, Wisconsin, and we have new ones in New York and Southern Ontario. The same set of about 50 clones replicated across these regions. So what we're doing now over the next few years, we're collecting data on these clones. Really, what's exciting to me is there's two things. We can find locally adapted hybrid selections. But what I'm really searching for are those that show wide adaptation. So we may find some top performers that actually perform well, it's my hope, across multiple regions. And this is, of course, being exposed to the pathogen, which is diverse across the range here. So hopefully looking for those that are durably resistant, but also in one sense, maybe climatically resilient. If you can grow in Nebraska, you can grow in New Jersey, and you can grow in Wisconsin and Minnesota, there's a chance that when the climate throws something weird at you, we've identified some of these plants that can handle it. And the early tests are showing that, so we're excited over that. And of course, we're going to use this information to continue the breeding program, because this is where we're going to learn about adaptation and resilience and durable disease resistance, and use a lot of the tools that Jeanne has mentioned in support of the breeding program. But I'll end here. The future does look bright for hybrid hazelnut production in colder regions. Like I mentioned, we joined forces with the Upper Midwest Hazelnut Development Initiative, and we have this opens up opportunities for sharing plant material even wider, although we've been doing that for a while, and learning from each other as we go. We're studying inheritance of eastern filbert blight resistance, and we'll be studying all of these other traits related to adaptation, phenology of bloom, cold damage, cold tolerance, as we go forward. So I'm just going to end there. I hope I wasn't keeping track of time very well. So hopefully we're okay. And I'll
let you run a little bit there, Tom. But
okay, I'm sorry.
We should get to a few questions. I bet Jeanne is pretty pleased about that, though, because now you're the one, you went the longest, and she didn't go the longest. So there was one. Yeah, see, I knew she'd be happy about that. So we do have some interesting questions. Thanks to Jeanne and Emily for answering some pertaining to their talk as they went in the chat. So if you saw some of those questions and now they've disappeared, you can click in the answered section and look at those. One question that came up, Tom, and we'll try to move on from this one quickly, is why the hazelnut fungus is such a problem now. Has it always been, or are there other ecological changes that occurred? I think the question comes in the context, if the hazelnut is native, why is it a big problem now? So clarity there.
Oh, well, real quick, I think that it's only been a problem when we've really tried to grow the European hazelnut. If you go back to the records from the early or the late 1800s, that's where it got first documented, when we tried to grow clones from Europe. And as breeders, we try to find resistance genes, and sometimes those are single gene resistances, and the fungus is actually able to overcome them and challenge them. So I think you're going to see more of that as breeders move forward with plants. We're trying to combat that with different breeding approaches, and maybe we'll discuss some of that. But the fungus has always been here, and we've really not grown European hazelnuts in the eastern United States. And if we have, they've been sort of a short-term crop, maybe 5 or 10 years, and then the fungus finds its way and destroys the orchards.
So we have a question that just came in here, and then I'll jump to a question on durable resistance to eastern filbert blight, and maybe everybody can jump in on that one. So this one might be interesting to Emily as well. So is there much need for expanding collections of Corylus americana? How well is the native range represented in current breeding repositories? And these repositories might not have been represented in the figure that Emily presented from the Curie article earlier.
If I could just mention real quick, I failed to put it as a slide, but we have been collecting American hazelnut germplasm for the past 10 years or so, and we have a collection that's over 2,000 accessions. It's actually too large for us to manage at this point, but from a wide representation across the native range. The southern regions are a little bit lacking, but we have a considerable collection, and when you combine the Oregon states, it's deciding what to do with it, how to manage it. That's also a big challenge.
Yeah, maybe ironically, I think one of the gaps is Missouri, for what that's worth. There are a lot of plants out there, but we need to go collect some. So if we could go to durable resistance, since we have just a few minutes, and maybe this would pertain to everybody. What does that look like in the hazelnut crossing scheme, Tom? So Jeanne has mentioned that single gene resistance is obviously less durable than quantitative resistance, but what are the approaches for breeding durable hazelnut?
So that's a great question. So if we look at the European hazelnut, one of the great things that really Sean Mellenbacher has spearheaded, but we've helped a little bit, is mapping resistance genes. We now have about at least 80 different sources of resistance. And when I say sources, these are from germplasm collections where we found resistant individuals. That's great and all, but the next step is trying to figure out why are they resistant? Is it one gene that's protecting them? Is it multiple genes? We don't know. So we've been mapping resistance genes and basically finding their location on different chromosomes. But as Jeanne sort of mentioned, there's still a black box there. We don't know what they're doing. And just because you're in a different chromosome, you don't really know that you're a different resistance gene or resistance allele. But we look at inheritance patterns and we can see different patterns and we can say, well, this is probably different. Now that we have genes mapped to different, what we call linkage groups, we can start making crosses and in theory, pyramiding these major resistance genes. And over the past 5 or 6 years, that's been one of our major approaches, is to make crosses between plants with known resistance genes on different chromosomes. And now we have molecular markers to help us sort that all out in the end, or developing them. So we're using gene pyramiding, and we've identified plants that what we consider carrying quantitative resistance, meaning they're not fully resistant. They have reduced disease incident. They have smaller cankers. They have less cankers. We've actually found when we cross unrelated quantitative resistant parents, we get a big improvement in the offspring, or we'll get that bell-shaped curve, and we're able to select on the farther side and get plants with what we would call resistant phenotypes from tolerant parents. So we've really been focused on quantitative resistance, our gene pyramiding, and now it's more complicated, as Jeanne can add to it, embedding single genes into the quantitative resistance background. And I'm not even going to mention working with American hazelnut, which is multigenic, confusing, challenging. But we're also on a whole another side of the breeding program looking at resistance from American hazelnut. So as a breeder, I learned early to just have a wide foundation in approaches, because some of them are not going to work. Many of them may not work.
So you're not going to mention it, but I think it's interesting to mention. So, and maybe this is where Jeanne could or would be compelled to chime in. But so what would be some of the challenges? Because if you're trying to pyramid like a major quantitative resistance loci and a single gene, if you have a single gene, you're not going to have a quantitative resistance background expressed. So you can't really do that with just phenotyping, right?
Well, can you hear me?
Mm-hmm.
Okay, this is a problem with gene pyramiding. So how people are trying to do it now is we now have technology that allows us to genotype 70,000 different places in the genome, and we can find individual little bits of DNA that are very, very close to where we believe, to where the data suggests that all these genes are. So you follow the DNA markers to incorporate the resistance, but there is a risk in this because your DNA markers are probably not in the gene where the action is. We don't know where the action is. I mean, the basis of disease resistance is just an undiscovered country. Well, we're chipping away at it. So by using a very clever molecular marker strategy, it is possible to do this pyramiding. But even when you've done that, you don't necessarily know. This doesn't guarantee success. I mean, you have to plant out the plants and do the phenotyping. There is no way you're ever going to get rid of that, nor should you, because that's what's going to be in the grower's orchard. So this approach, gene pyramiding, can work very well in highly characterized systems with a short generation time. It is my belief it will also work in perennial crops. We just have to be able to have patience and also keep a lot of things on the burners. So you just don't do one cross, right, Tom? You do a whole lot of crosses and you do a whole lot of screening, knowing that you won't be right. You won't be successful every time, but you'll be successful enough times so that you can take more steps towards durable resistance. I mean, we're trying to recapitulate here in the course of a career or two what Mother Nature has done over tens of thousands of years, or even hundreds of thousands of years. I don't think that's arrogant. I think it's possible. It just takes a realistic understanding of what you can achieve, and a couple of big winners to keep people funding the program.
So, yeah, I was wanting to connect, and maybe you address this a little bit. The maintenance of genetic diversity, because that was something that we talked about earlier on, with the concept that could seem somewhat at odds with that of applying selection pressure and finding these, unique recombinants that have all the commercial characters as well as the multiple pyramided loci. So that does seem like a pretty tight bottleneck. How do you reconcile that with maintenance of diversity, or even not releasing single clones, but maybe a small collection of clones? So the recent releases from Rutgers were called a series, so maybe that steps towards us a little bit, but it's probably a part of the equation, not the whole equation.
One of the saving graces in hazelnut is you need a certain size for a certain application. You want it to taste good. You want the shell to be thin, but it's not like some other fruit and nut crops where it's got to look a certain way that has sometimes not much to do with quality. That greatly complicates your life. So these goals are achievable, and it has to do with using a wide base of parents. Now, like Tom says, I mean, all this takes an immense amount of land and space, and an individual program is limited by the amount of space, for example, that Rutgers is willing to give to this crazy project, right? I mean, a lot of us work in projects that why are you trying to incorporate emerald ash borer resistance into ash? Just let them all die. We'll plant something else. Why are you doing this, Tom Molnar? What does it matter whether we have a hazelnut industry in the eastern United States? The long-term goal of permaculture is to have some permas to culture. And it's this work that does that while at the same time paying attention to genetic diversity and local adaptation. The amazing thing is when you look at genetic diversity in our cultivated perennial crops, grapes, apples, grapes and apples specifically, there is a high amount of genetic diversity in the cultivars, which is surprising. But over the course of time, a high level of genetic diversity has been maintained. And that was alluded to by was it you, Emily?
Yeah. And I just want to jump into and mention, Jeanne, you're absolutely right that, no one institution is going to be able to maintain the diversity of these collections. And that is really kind of what we're trying to get at in part with this consortium to conserve North American fruit and nut trees, because we saw that gap there. And so by bringing together different institutions and maintaining collections at different sites, that's one way we can start to address that problem.
Yeah, I think that's a great, and I think it's a great way for underutilized collaborative opportunities between the botanic garden systems and public universities, which is something that who was active in the chat, Mark Cogshaw, harps on all the time. So I'm sure he'll be happy that that's getting mention of. So we're a few minutes past when Hannah wanted to make some closing remarks. So I might
That's totally fine. I don't need much time. This is great.
Okay. Well, I've already put my foot in my mouth, so I'll just say thank you to the panel. I really appreciated the discussion and the time, especially Emily, who's at a conference as we speak, so she's doing double duty. And I'll turn it over to Hannah.
Great. Thank you all so much. That was just a perfect closing, I think, to this day. And I've been adding some quips and just highlights from this session and all of them into the discussion board, so you can check that out. Presenters, especially if you can stick around to answer some of the questions that are left in the Q&A, that would be really great. But I am just going to close by saying thank you all for being with us today. If nothing else, I hope these presentations have left everyone with an expanded appreciation for all that goes into perennial crop improvement and integration and the huge importance of all aspects of this work. So thank you again to the presenters, to everyone who has been participating in the lively discussion on these topics. And if you missed any sessions, not to worry, you will receive notice as soon as the symposium recordings are available on our YouTube channel and archived on our website, along with the discussion board. So thank you all and have a wonderful evening.